Good morning, students. Today's topic is dihybrid experiment. I just mentioned it. Dihybrid experiment. What is dihybrid experiment? We are studying right now the Mendel's principle of inheritance. जीवों में सेक्सुअल रिप्रोडक्शन में कैरेक्टर का ट्रांसमिशन कैसे होता है? The pattern of transmission of characteristic is called inheritance. And we are studying right now Mendel's principle of inheritance. और इस प्रसंग में, in this context, we have studied monohybrid experiment in our last class. So, monohybrid experiments was the experiment conducted by Mendel to study the inheritance of single characteristics. The motto of the experiment was to study the inheritance of single characteristics. That means single characteristics was under watch. इसका मतलब यह नहीं कि उस प्रोडक्शन में एक ही कैरेक्टरिस्टिक ट्रांसफर हो रहा है. ट्रांसफर तो सारे हो रहे हैं, लेकिन हमारा उद्देश्य था एक गुण को स्टडी करना. One characteristic that may be controlled by one gene, so that is also called the inheritance of single. अब हमारा उद्देश्य होगा कि हम दो गुणों का अवलोकन करें, अध्ययन करें, to study the inheritance of two characteristics along एक साथ, and this is called dihybrid experiment. So the experiments conducted by Mendel to study the inheritance of two characteristics along is called dihybrid experiment. So Mendel selected two characteristics in one pure live breed. For example, the seed shape and seed colors in a pure line was taken as characteristics. That was seed shape was round seed and yellow in color. Round and yellow. And the other seed was, the shape was ring-colored and the color was grey. So he selected two pure line entities, and as we know that the result in F1 generation will demarcate which one is dominant and which one is recessive. So when we cross these two pure line bricks, hello? Yes. I just call you in 2 minutes. I'm in class. Okay. 2 minutes. 2 minutes. ठीक. Then when he crossed yellow with green one, then the entire progeny was round and yellow. Round and yellow. That means the shape round and the color yellow is dominant. So we can mark here capital R and capital R for round shape, and capital Y and capital Y for yellow color. Whereas the small r and the small r will denote the recessive allele, whereas for green a small y and a small y will denote. This is F1 generation. When the Mendel left this for self-pollination, then what will be the genotype here? It will be R capital, R small, Y capital and Y small. This is the genotype. Now, when the self-pollination will occur, what will be the gametes? The gamete will be of 4 types. R capital, Y capital, R capital, Y small, R small, Y capital, and R small, Y small. This will be the gamete for F2 generation. In F1 generation, F1 generation will produce this gametes in male as well as female. So what will be the probability of progeny? That we will find with Punnett square box. So R capital, Y capital, R capital, Y capital, female, male, then R capital, Y small, then R small, Y capital, then R small, Y small. Here R capital, Y small. Here R small, Y capital. Here R small, Y small. So this is Punnett square box, and it will present the progeny of F2 generation. And on the basis of this Punnett square box, we will find out the phenotypic ratio as well as genotypic ratio. So, what will happen if R capital Y capital will fuse with female gamete R capital Y capital? Here R capital, R capital, Y capital, Y capital. Here R capital, Y capital, Y small. Here R capital, R small, Y capital, Y capital. Here R capital, R small, Y capital, Y small. Here R capital, Y capital, Y small. Here R capital, Y small, Y small. Here R capital, R small, Y capital, Y small. Here R capital, R small, Y small, Y small. Here R capital, R small, Y capital, Y capital. Here R capital, R small, Y capital, Y small. Here R capital, R small, Y capital, Y capital. Here R small, Y capital, Y small. Here R capital, R small, Y capital, Y small. Here R capital, R small, Y small, Y small. Here R small, Y capital, Y small. Here R small, Y small, Y small. So this is the phenotype. It will be more clear when we will present it with color combination. Here the shape will be round, color will be yellow. Here the shape will be round, color will be yellow. Here the shape will be round, color will be yellow. Round, yellow. Round, yellow. Round, but green. Here the color will be round but green. Here round yellow. Here round green. Round green. Here also round green. Here round yellow. Round yellow. Here round yellow. Here a wrinkled yellow. Here a wrinkled yellow. Here a wrinkled yellow. And here a wrinkled. So what do we find in F2 generation? We just shift it little more this side. That was wrinkled green. This was the round yellow. Here produces all round yellow. Phenotype now here the combination genotype is R capital, Y capital, Y capital. Here R small, Y small, Y small. Here it is R capital, R small, Y capital, Y small. This is F1 generation. And in F1 generation, the gametes are 4 types, which is mentioned there. R capital, Y capital, R capital, Y small, Y capital, and R small gametes will be formed of 4 types. And on the basis of that, in F2 generation, what is this? F2 generation. Phenotypic ratio. Phenotypic ratio. Round yellow. Round yellow. Round green. Round green. Wrinkled yellow. Wrinkled yellow. Wrinkled blue. This is 9, this is 3, this is 3, this is 1. So phenotypic ratio is equal to 9:3:3:1. This 2 is recombinant one. Recombinant. Round green, round and yellow, round and yellow. The parental type was round and yellow, and the wrinkled and green. So the parental has combination of round yellow, but here it is round green. The parental has combination wrinkled green, but it is here wrinkled green. So there are 2 types of recombinant which comes in the F2 generation. So in F2 generation, the phenotypic ratio is 9:3:3:1. Whereas the genotypic ratio calculation is somehow different, as we will have to find out what is the number of a specific genotype in the progeny. So this part is clear. So we will rub it to focus upon the F2 generation. So genotypic ratio. Genotypic ratio is equal to R capital, Y capital, Y capital. This is the parental type and its number is 1. So this is clear. Then comes R capital, Y capital, Y small. Its number is 2. 1, 2. So it is 2. Then comes R capital, R small, Y capital, Y capital. R capital, R small, R capital, R small. Its number is 2. So it is also 2. Now comes R capital, R small, but before that R capital, Y small, Y small. And its number is 1 here. So it is 1. Now R capital, R small, Y capital, Y small. Its number is 4. 1, 2, 3, 4. So it is 4. Now R small, Y capital is equal to 1. It is here. Now R capital, R small, Y small, its number is 2. So 1, 2. Then R small, Y capital, Y small. Its number is 2 also. Now R small, Y small, Y small. Its number is 1. So when we will mention the genotypic ratio, we will write each genotype, then its number. So genotypic ratio is equal to R capital, Y capital, Y capital is to R capital, Y small is to R capital, R small, Y capital is to R capital, Y small is to R capital, R small, Y small is to R small, Y capital is to R capital, R small, Y small is to R small, Y capital, Y small is to R small, Y small is equal to 1 is to 2 is to 2 is to 1 is to 4 is to 1 is to 2 is to 2 is to 1. This is the genotypic ratio. Genotypic ratio. So this is dihybrid experiment of Mendel, in which F2 generation have phenotypic ratio 9:3:3:1 and genotypic ratio is R capital, Y small is to R capital, R small, Y capital is to R capital, Y small is to R capital, R small, Y small is to R small, Y capital is to R capital, R small, Y small is to R small, Y small is to R small, Y small. 1 is to 2 is to 2 is to 1 is to 4 is to 1 is to 2 is to 2 is to 1. That means ये phenotypic ratio 9 जो है वो डोमिनेंट आया और पेरेंटल फॉर्म में आया. रिकॉम्बिनेंट भी है तो पेरेंटल यूनिट में है. और एक जो ये रिसेसिव कैरेक्टर आया ये भी पेरेंटल फॉर्म में आया बट रिसेसिव. और ये 3:3 जो है इस 9 पेरेंटल टाइप में भी एक जो है वो प्योर है और बाकी के 8 2 2 4 जो है 9 3 12 3 16 तो जो 8 है इसमें उस 8 में 7 जो है वो रिकॉम्बिनेंट है. 1 2 3 4 5 6 7 8 9. इस 9 में इस एक को छोड़ के बाकी जितने भी टॉल हैं, रेड है, राउंड है और येलो है, वो सबके सब रिकॉम्बिनेंट है. और ये दोनों जो है ऐसे रिकॉम्बिनेंट हैं जो पेरेंटल टाइप से भी अलग कॉम्बिनेशन जिसमें दोनों कैरेक्टर का अलग अलग दिखा, पेरेंटल टाइप से अलग दिखा. ये रिकॉम्बिनेंट तो है इस 9 में 8 जो है वो रिकॉम्बिनेंट है, लेकिन ऐसा रिकॉम्बिनेंट है जो F1 जनरेशन की तरह है और जो दो कैरेक्टर पेरेंटल टाइप में जो थे वो एक साथ थे. लेकिन ये दोनों ऐसे रिकॉम्बिनेंट है 3:3 जो पेरेंटल कॉम्बिनेशन नहीं है. यानी कोई भी दो कैरेक्टर जो एक जीव में हमेशा साथ में अभी है इस जनरेशन में, वो नेक्स्ट जनरेशन में भी साथ में रहेगा ये जरूरी नहीं है. एंड दिस इज कॉल्ड लॉ ऑफ we can say that on the basis of dihybrid experiment, Mendel formulated his third postulate, which is called law of independent assortment. So we have understood dihybrid experiment. Now we will discuss the law of independent assortment in next class in detail. But we just summarize it that the parental combination was round and yellow. Round and yellow was parental combination. Wrinkled and green was parental combination. But these two appeared in F2 generation, which is not the parental combination, where the characters does not remain alone. Round was associated with yellow color, but here round came with green color of shape. And here wrinkled was associated with green color, which comes with yellow. So this is called law of independent assortment. So this is all about Mendel's dihybrid experiment. If you have any doubt, please contact me on my WhatsApp number. Now we will meet in next class. Till then, goodbye.